Next biomolecule is protein. The building block, the monomers of proteins is amino acids. So the proteins are made of amino acid. There are totally 20 different kind of amino acid. And amino acid is called amino acid because they have the amino group. This is amino group. And there's the carbolic acid group. So it's called amino acid because it has these two functional group. And when you have an amino acid, you have and another amino acid, you connect them together. It's called dehydration synthesis because you remove a OH, remove a H, and now they form the covalent bond together. But for protein, this covalent bond has a unique name called peptide bond. Because it always on one side here is a CO, the other has an NH. So this covalent bond is called a peptide bond. It's a very unique one. Peptide bond only exists in protein in the amino acid sequence. It does not exist in other biomolecules. And this is amino acid structure, amino group, carbolic acid group. And there are totally 20 different kind of amino acid. So in these two groups, they're always the same. And this is called R group. This one changed. There are totally 20 different kind of R groups. So different amino acid like alanines, leucines, and all this have different kind of R group. And these R groups decide the amino acids, their characteristic. So there are 20 different kind of them. And some functional group, they don't like water, so they become the hydrophobic amino acid. And when they form a protein are made of amino acids. These amino acids will try to hide inside because they don't like water. And in the human body, it's all water. And you also have the amino acid that hydrophilic, means this functional group, they like water. So when they form an amino acid, when they form the protein, which is made of amino acid, and this functional group have no problem touch water. So they're probably gonna face outside. And these are the polar one, no specific, no net charge. So they have the hydrophilic, hydrophobic, or neutral one. So totally 20 different amino acids. You don't need to know which one is which. That's not the point of this class. You just know, okay, proteins are made of amino acid. There are 20 of them, and some of them hydrophilic, some of them hydrophobic. And some amino acids, they are called essential amino acids. The reason is they can only be get from food. So our body can produce some amino acids, not all 20. We can produce some. They are called non-essential. But these 8, you have to get them from food. So it's called essential amino acids. And when you have amino acid, 2 of them, it's called a dehydration synthesis. You remove a OH, remove a H, they form a peptide bond. Okay, now you combine these 2 amino acids together. And when you eat meat, when you eat steak, okay, well, your digestive system will cut this. It's called hydrolysis, become a single amino acid. So your body can use those single amino acids after you eat steak, eat chicken, and then use them to build your own protein. So they form the peptide bond. Again, peptide bond can only be found in protein, in amino acid, this kind of biomolecule. So the monomer is amino acid. Okay, now you combine them together by using peptide bond. First, this is called the polypeptide or amino acid sequence. This is not a protein yet because protein need to have its correct 3D structure to perform its function. So now you have an amino acid sequence. This is called a 1D structure. Now you're gonna take the 1D structure to fold become a 2D structure. And the most famous two 2D structures are called alpha helix and the beta sheet. So both of them are the 2D structures of the protein. And after you have the 2D structure, and this 2D structure are mainly connected, formed by the hydrogen bond. You can see a lot of dashed line here and a lot of dashed line here. So alpha helix, beta sheet, these two are the 2D structures of protein. And they are connected by peptide bond, by hydrogen bond. Now you have the 2D structure. You're going to use a lot of 2D structure to form the 3D structure. Every protein at least have a 3D structure to perform its function. When you damage this 3D structure, and the protein could not perform its function, and it's called denature. And two ways to denature the protein. One is pH, the second one is temperature. So we talk about denature proteins. And some proteins, they have 4D structure. This is the textbook example called hemoglobin. Hemoglobin is a protein inside your red blood cell. Its main function is to carry oxygen. And you can find there are 4 3D structure combined together, and each one, the center is iron. So if you have anemia, which means not enough red blood cell or not enough hemoglobin, the doctor will prescribe the iron pill. The reason is the center, this red one is iron, and it binds with oxygen. So each hemoglobin is made of 4 3D structures. So you actually have 4 oxygen binding sites. And some proteins, especially they are big, they can even have 50, 60 structure, but at least they need to have 3D structure. So the primary structure is called the primary amino acid sequence. And then you have a 2D secondary, and the 3D is called the motif. And some proteins has a 4D, 50, even 60. Depends on the protein. Some proteins are big, others are small. So let's look at the bond, connect them together. First one, to maintain the 3D structure. First one is hydrogen bond. You find in 2D, from 2D, you have a hydrogen bond. And the second way to connect the proteins, maintain structure, is the disulfide bridge. And you can maintain 3D structure. Then the most important two are these two. Number one is hydrogen bond. Number two is disulfide bridge. And then you can have ionic bond or those amino acid hydrophobic, hydrophilic structure. So it's called hydrophobic exclusion. Apparently, when these amino acids, they don't like water, they will be folded inside. So they will decide the protein 3D structure. And a protein, for the protein to work properly, it need to maintain its correct 3D structure. When the 3D structure could not be maintained, it could not perform its function. So every protein need to have its correct 3D structure. And some protein are very big. Lactase, this is an enzyme digest lactose. And protein 3D structure is decided by 1D because it's made of amino acid. It's made of amino acid. And some amino acids are hydrophilic, others are hydrophobic, or they are neutral. And once you have a point mutation, we'll talk about the transcription translation later. It comes from the DNA. So when the DNA sequence is wrong, the RNA is wrong. When the RNA is wrong, the amino acid is wrong. And say we have one amino acid is being replaced. It's called a point mutation. And these amino acid, they have different characteristics. Some hydrophilic, other hydrophobic. And when you replace the wrong one, and the result is it will fold incorrectly. And the protein's 3D structure been changed. And when it's been changed, it could not perform its proper function. This is called the sickle cell anemia. These red blood cells, they're supposed to look like a donut. So this helps them to carry oxygen through the small capillary. We have the capillary, their inner diameter is like the size of red blood cell. And when they turn into this shape, it's more difficult for them to carry oxygen. So it's anemia. Anemia means you don't have enough red blood cell or you don't have healthy red blood cell. That's anemia. The person's still alive, so they can pass their gene to next generation. That's why we still have the sickle cell anemia. So what happens is the red blood cell look like strange shape. And when they start to carry oxygen to through the small capillary, they can't. So their oxygen carry ability is being compromised. And protein can be denatured by temperature. It can also be denatured by pH, change the pH. So denature means it changes 3D structure. It's 1D is still correct. So you can eat a fried egg. You still have all the nutrients from the egg. It just does not have its correct 3D structure. It's irreversible. So you could not cool the temperature down. The fried egg won't go back to the original egg. So it's irreversible. And its 1D structure still the same amino acid. So you eat a fried egg, you eat a raw egg, you have the same nutrients. And proteins has a lot of function because the whole body is made of protein. So proteins like these are the muscle. When you learn AP1, you will learn this called the sarcomere. That's the functional unit of the muscle. They will become shorter. That's how the muscle contract. So proteins function, it can provide movement. It can transport, like this is the hemoglobin. This is the protein inside your red blood cell. It can transport. And some proteins are located on the cell membrane. Its function is transporter because some molecules, they need to go in, go out of the cell, and they could not go through the cell membrane. Cell membrane is mainly made of phospholipid, especially if the molecule is not lipid soluble. It's difficult to go through the cell membrane. And if the cell need them, they need to use the channel or carrier. We work the transport proteins to make the ions move in or make them move out. And some of them require ATP. So we'll have a separate lecture talk about those transporter protein. And protein can work as a buffer because it's amino group, it's carbonic acid group, so it can take hydrogen or it can release hydrogen. That's buffer's function. So it can maintain the hydrogen concentration inside your body. So it can maintain the pH. And some proteins, they can work as receptor. Receptor is on the cell membrane. So the signals, like the hormones, need to bind with the receptor. So like insulin's job is to bind with the insulin receptor. And once they bind with insulin receptor, its function is will put more glucose carrier. Glucose could not directly go through the cell membrane. So glucose have to go through the glucose carrier in order to go into the cell. And after you eat a big meal, your body release insulin. Insulin's job is to trigger bind with insulin receptor and put more glucose carrier on the cell membrane. And the result is more glucose can be transported in. So your blood sugar level decrease. So that's insulin's function. And if this response is not working well, it's called the insulin insensitivity. And when the person have serious insulin insensitivity, there's diabetes too. Diabetes is they could not maintain their blood sugar. Okay, and protein can work as a hormone. We have peptide hormone in the body. So these are made of proteins. And proteins can work as defense function, like an antibody is also a protein. And protein can work as an enzyme because enzymes are proteins. And we know enzymes have specific binding site. So for a specific substrate. So each enzyme can only digest a specific molecule. So far, all these are the proteins function. They have a lot of function because the whole body are made of proteins. Okay, let's stop here.